This is our mitosis paper slideshow by Jake Ryer, Jared Kalinsky, Connor Fergus, and Brett McCartney.
The first stage of mitosis is interphase. The cell spends most of its life in this stage. In this stage, the organelles double, DNA is replicated. Interphase is created of 3 stages. It is gap 1, synthesis, and gap 2. In gap 1, messenger RNA and proteins are created. In the second stage, synthesis, DNA is replicated. In the third stage, gap 2, the cell resumes growth and makes proteins to drive division.
Prophase is the next stage of mitosis. Chromosomes has condensed so much that they are visible under a light microscope. The duplicated chromosomes become visible as they condense also. One of the 2 centrosomes moves towards the opposite side of the cell as the nuclear envelope breaks up completely. Metaphase is the next stage. All the chromosomes are aligned midway between the spindle poles.
Anaphase is the third cell of mitosis. The spindle poles pull the sister chromatids away from each other towards opposite ends of the cell. The resulting daughter cells will have identical pairs of chromosomes. And telophase, in telophase, the chromosomes result in opposite ends of the cell. A nuclear envelope begins to separate DNA from the cytoplasm. The originally tightly packed chromosomes begin to loosen up, and this ends mitosis and starts cytokinesis.
Cytokinesis occurs at the end of mitosis. Mitosis is the mechanism of cytoplasmic division and the physical process of cell division. It divides the cytoplasm of a parent cell into 2 daughter cells. It separates the cell in half and ensures that one nucleus ends up in each daughter cell. In an animal cell, the first visible change is when the cleavage furrow is created on the cell's surface. The furrow deepens and spreads around the cell up until it fully divides in 2. But in plant cells, because they have a cell wall, no cleavage furrow can be formed. Instead, a cell plate forms across the cell where the old metaphase plate was. Eventually, the growing cell plate combines with the current plasma membrane, and this produces the 2 daughter cells. The end.